So now that we had a fun quiz session from the first slide, let's start with the asexual reproduction and how it works in prokaryotes. It's very easy, but it will also make a lot of things clear to you in the sense that all those techniques that you use in molecular biology, they have a natural origin. They're not invented, they were discovered. So all the techniques, right? Everything in this world, really believe me, in biology is a discovery that you discover that a certain organism uses a certain method and then we convert it into a technique and we make money out of it. True, right? So as we discussed in the last class, much simpler class, this one will be a little more difficult, still very easy for a lot of you, is the transfer of genetic material in prokaryotes. This transfer of genetic material is done by binary fission as shown on the right side of the slide. And here you see that the bacterial chromosome, right, there's no nucleus, right, is present in a bacteria. The replication takes place of this chromosome and the mother cell pinches off into daughter cells and divides into two cells. This is why it's called bi or two, right? Binary fission, right? So this is the main method, but of course this takes some kind of methodology for the prokaryotes, which we have labeled as, right, all these techniques, transformation, conjugation and transduction, right? So we studied them during molecular biology. Let's discuss these techniques from the point of view of how bacterial cell uses them. What is the natural origin, the base of these techniques? So the first one, which is called transformation, if you remember, we were introducing naked DNA into the cell. The same thing. The transformation occurs when prokaryotes acquire genes from their surrounding environment. This is a normal process that is happening, right? And where does this foreign DNA come from? It is either left behind by other bacteria, which could be from the same or different species when they died, right? So it could be just because the bacteria died, and this DNA can be from the same species or the different species. So this DNA, which is shown as the plasmid in this image on the right side, right, because we are still kind of connecting it with molecular biology, this foreign piece of DNA is then directly taken up by the cell. It's a naked piece of DNA and is expressed. So I'll give you two examples of what happens in nature to help you out in understanding this. And that is how it became the basis of molecular biology. The first one is that, let's say the bacteria encountered a piece of foreign DNA. If this foreign DNA is beneficial, which means that it gives some kind of success or some kind of advantage to this bacteria. For example, it codes an antibiotic resistance, right? It is a known fact that bacteria want to save themselves from other bacteria, and for this they have antibiotic resistance. Some of them have antibiotic resistance to the antibiotic that is produced by the other bacteria or other source, right? So let's say that the foreign DNA has this beneficial gene of antibiotic resistance, and any individual bacteria that would have this would get a selective advantage, right? Because let's say one bacteria picked this up but the other bacteria did not, and then they countered this antibiotic, So this bacteria would be at advantage, right? So as long as the individual with this gene reproduces successfully, which also means that this gene sticks around in the cell without removing this gene, it will be more fit, right? And this can be called as microevolution, right? Via natural selection. So this is an example of an advantage. Something else can also happen. That is transformation of a non-pathogenic. Non-pathogenic means, right? It means that the bacteria which does not cause any pathogenesis or a disease, mostly we are talking about us humans, right? Animals. How would this happen? For example, Streptococcus pneumoniae, right? If they are placed in a medium where you have dead cells of a pathogenic strain, these Streptococcus pneumoniae can take up the DNA from the dead pathogenic cells and they can become pathogenic. So this would be a disadvantage to us, right? Right? In general, it would be a disadvantage that the non-pathogenic strain converts into pathogenic strain. So these are two very known examples of transformation in nature, what happens. And of course, we have discussed it so many times that transformation is a common technique to relocate bacterial genes which we use in molecular biology. The next method is transduction. And how did we do transduction? Through viruses or phages. And the exchange of DNA between prokaryotes is therefore made possible. So this is shown in the picture on the right side. You see a phage, it attaches, releases its DNA into the bacteria. Then the DNA reproduces, right? Let's say what do you call it? Transcribes and translates and makes more phage molecules which are released. Now, what is the most common or recent example? COVID-19, right? This is how it is reproducing through transduction, right? It is attaching itself to the membrane of our cells through spike proteins, remember? Then it enters the cell, releases the positive strand mRNA, right? Which is then transcribed and translated, makes the viral molecule again and releases it outside the cell. Remember that diagram? You have studied it for your midterm exams too. Okay, so in specialized transduction, this is just an example for you. The phage DNA inserts somewhat benignly. Benignly means it's not active. It just inserts its DNA into the bacterial host chromosome. Right? So it is inserted into the chromosome. Here it can lay dormant for many generations. This is a very common thing in nature. That the phage DNA is inserted and then it is not used up in many generations of bacteria. But remember, bacteria do not survive for 80 years. They are doing it 20 to 3 hours, 20 minutes to 3 hours, remember? So their many generations are over much more before our many generations, right? However, under certain conditions, so it can happen that the phage inserted the DNA but nothing happened. But some kind of environmental changes result in the phage DNA getting excised. Excised means out of the bacterial chromosome, right? Where it was inserted. This causes it to replicate and form new phages that burst out of the cell. These phages can reinfect other bacteria and thereby transfer not only their own DNA but pieces of the former host DNA. So this will be more clear in this diagram here, right? So you see that a phage is attaching to a host cell on the top of the slide and injects the DNA. So the white piece is the phage DNA. This DNA, then two things can happen, right? First is the lysogenic cycle, which is on the right side, where this DNA gets inserted into the chromosome. Now, why is this important? This is important because once it is part of the bacterial chromosome, then it can survive many generations of binary fission. If it will not be part of the bacterial chromosome, it will not be replicated in any binary fission. So this is why this is important. This is called as the lysogenic cycle, right? So in this way, the phage DNA can stay, right, in the bacteria molecule. What can happen at the end of this is that the prophage, because now it is called the prophage, it is not viral or how do you say pathogenic, it is part of bacterial chromosome, so it's called prophage. It can exit the host DNA at a certain point, right, and enter the lytic cycle, which means it is a separate phage DNA again. When this happens, the phage DNA hosts the machinery of the host cell to produce copies of the phage, which is shown in the lytic cycle on the left, lyses the cell because now it kills the cell to get out, and release. This can infect more cells. So these are the two methods in the nature through which transduction works, right? And this could be now hypothetically this could be one of the reason how viruses survive in nature, or even for example in your body where you get recurrent infections, because remember our body has 3 million bacterial cells, right? The last one on the list is conjugation. So the mechanism, you know this, there's a formation of a conjugation tube. We call it conjugation tube in molecular biology and sex pilus in natural condition, it is called so, right? So formation of a conjugation tube and transfer of the DNA. So this is called a pilus, or and usually the pilus is only present on the male, right, male bacteria we call them. Of course it is very difficult for us to understand how it is male bacteria, but this is how they are called. In order to produce these pili, prokaryotes must have a plasmid termed the F factor. So this is how we define the male, right? The male bacteria has the F factor or the fertility factor plasmid, right? And that is how it can form this pilus to transfer the DNA. Now some trivia for you. When a cell has the F factor plasmid, it is called F positive. This is a very GPAT relevant question. So what is F positive? How is a bacteria F positive, for example? So any bacteria which has F factor plasmid is called F positive, and it is heritable. What does that mean? That means that if an F positive cell divides, both of the resulting cell will be F positive, right? Binary fission. And this condition is contagious, which means that after an F positive cell conjugates with a female cell, it gives the F factor plasmid to it, and therefore the female cell also becomes male, right? So when the F plus cell transfers the plasmid to the female, meaning F negative cell, then this F negative cell also becomes F positive. So this is how conjugation works. Now again, I would like to remind you that group 1, your assignment is due, right? And you must do this right after your midterms. If you have any questions, we can discuss about this in the tutorial. So please come to the tutorial, right? And anyways, I will be taking tutorial to answer any questions about this part or the other part which comes in your midterm examination. Okay, then have a great day and keep safe. It's not very safe nowadays.